Hello, this is Dr. Liz Reuter, and today I'm going to be talking with you a little bit about nutritional genomics. The objectives of the presentation today are to help you gain an increased knowledge of the concept of nutritional genomics, to better understand some basic genetic principles, and to help understand the relevance of nutritional genomics to health, disease treatment, and prevention. Nutritional genomics is still in its infancy, but it does have great potential for treatment and prevention of chronic diseases. The impetus for the shift to integrating genetic principles into healthcare began with the advent of the Human Genome Project. And this project began in the year 1990, and it was a multinational effort. The initial goals of the project were to identify all of the 20,000 to 25,000 genes in human DNA, and I will say we now know that there are about 25,000 genes, to help determine the sequences of the 3 billion chemical base pairs that make up human DNA, to put all that information into databases where it would be publicly accessible for no cost to scientists and the public, to help improve the tools for data analysis, and to make a framework that would address some of the ethical, legal, and social issues that may arise from the project. So I'd like to go over some definitions. You've probably heard many of these definitions before, but it can't help for us to review. So the genome is the total of an organism's genetic information. All living organisms have a genome, and today we'll be talking specifically about the human genome. DNA is deoxyribonucleic acid. This is the hereditary material in humans and in most other organisms. Almost all of DNA is found in the cell's nucleus, although there is a small amount of DNA found in the mitochondria, and this is referred to as mDNA. But today I'm going to be talking exclusively about the DNA found in the nucleus. So information on DNA is stored as a code of nucleotides, and we'll talk specifically about nucleotides in a moment. So overall, you can just think of the DNA as the carrier of the genetic information. RNA is ribonucleic acid. This carries the genetic code from the DNA in the nucleus to ribosomes in the cytoplasm where protein translation occurs. So the DNA is coding, it's giving instructions on what protein to build, but that protein is not formed in the nucleus. Instead, mRNA is formed in the nucleus. The term that we use is translated. So the DNA is translated into RNA. And then that RNA will travel out of the nucleus into the cytoplasm to a ribosome, and that is where the protein manufacture actually occurs. So the nucleotides are the building blocks of both DNA and RNA. Both DNA and RNA have a ribose phosphorus backbone. DNA has two backbones, and that's how those two backbones together form that double helix. RNA just has one, and both DNA and RNA have nitrogen-containing purine or pyrimidine bases. We'll talk more about that later. But in the DNA, it's adenine, guanine, cytosine, and thymine, whereas in RNA, it's adenine, cytosine, guanine, and uracil. So in the RNA, one of those nucleotides, the thymine, is subbed out for a uracil. So some other important definitions are nutritional genomics. This is a broad term that's referring to the field of study that describes the application of genetic technology to food and nutrition. So the term nutritional genomics is referring to both nutrigenetics and nutrigenomics. So nutrigenetics is specifically how an individual's genetic profile influences their requirement for specific nutrients or other bioactive substances. On the other hand, nutrigenomics is the study of how the nutrients and bioactive food components that we eat interact with the genome to influence gene expression. So in review, nutrigenetics is telling us our genes are kind of dictating our nutrient requirements. On the flip side, nutrigenomics is how what we eat is influencing the expression of specific genes.
But let's take a look here at this graphic. So nutrigenetics is how your genes are influencing your need for nutrients. On the other hand, nutrigenomics is telling us how nutrients, and this also includes bioactive food components, influence your genes. And I find the study of nutrigenomics really exciting. The ability of how what you're eating can influence your DNA. It's not changing your DNA, but it's influencing its ability to transcribe proteins. And we'll talk more about this later. All right, so let's spend a little time going through some of the genetic basics, which I know you've had before, but I think it's important just to kind of refresh ourselves since this might be something you haven't thought about in a while. So in the earlier diagram, the first thing we saw outside of that nucleus were the chromosomes. So now in these upcoming slides, I'm going to go through each of the terms that we saw in this previous diagram and talk about them in a little more detail. So the chromosomes are how large amounts of DNA are packaged in the nucleus. So each cell nucleus contains a complete set of genetic material divided among these chromosomes. Most individuals have 46 chromosomes in total. There are 22 pairs of autosomal chromosomes, so in total that would be 44, plus 2 sex chromosomes. And this could be an X and a Y chromosome for males, or it could be 2 X chromosomes for females. So again, each cell has a complete set of the genetic material. Doesn't matter if that cell is coming from your foot or your mouth or your liver. It has that individual's complete set of genetic material. In humans, as I said here, you've got 22 pairs of autosomal chromosomes, and that's pairs, right? So one set of the pair is coming from the father, one set is coming from the mother. And then along with these sex chromosomes, a male would have an X and a Y, a female would have 2 X chromosomes. And so the fact that men only have 1 X chromosome has implications for certain genetic conditions. And we'll talk a little bit more about that later. But as you can imagine, or as you may have learned previously, when there's a mutation in that X chromosome, and there's only one X chromosome, it's more likely then to present. So on the chromosome you have genes, and the gene is simply a sequence of nucleotides along the DNA strand that encodes the instructions for synthesizing proteins. So you can just think of this gene as an ordered sequence of nucleotides, and a gene is always going to be located in a particular position on a particular chromosome, and that's often referred to as the locus, the genetic locus, where that gene is located. So the code is embodied in the sequence of bases along that DNA strand. So a set of 3 bases is known as a codon, and that specifies one amino acid. So as you'll remember, amino acids are the building blocks of proteins. So you have these 3 sets of bases, which makes one amino acid, and then multiple amino acids in a sequence then makes a protein. So an average gene has about 3,000 nucleotides, and as I said earlier, the human genome contains about 25,000 genes. Interestingly though, genes account for about less than 10% of the total genome. The function of the rest of the roughly 90% of the genome is not known, and it's often referred to as quote-unquote junk DNA. And this might include things like intron sequences, which are defunct copies of genes, or long tracks of repeated sequences that have no known biological function. So it got this name junk DNA because at first people thought it really had no purpose at all. But now there's emerging evidence that suggests that some of this DNA might actually be involved in the regulation of gene expression. So it could be that some of this junk DNA is not really junk at all. But this is still an emerging line of research.
So let's take a closer look now at the structure of RNA and DNA. And we'll start first by talking about the DNA. So as I stated earlier, DNA is the genetic material of all living organisms. It is a double helix, and I don't think my pointer is showing up to you. Hopefully it is, but I'm looking over here on the right-hand side where it says DNA, and you can see this double helical structure. And this is formed by the base pairs, which you can see here in the center, that are attached to the backbone. So the backbone of the double helix is formed with a ribose and a phosphorus molecule. And there are 2 backbones here that form together to make this double helix. So there are 4 different base pairs in DNA: cytosine, guanine, adenine, and thymine. And the adenine will only bond to a thymine, and the cytosine will only bond to a guanine. And these, the nucleotides are bound to each other through a hydrogen bond. Sometimes these nucleotides are referred to as purines and pyrimidines, and this is referring to their chemical structure. So purines are the adenine and the guanine. So if you look at those structures of adenine and guanine, you'll notice that they both have 2 ring structures. And cytosine and thymine are pyrimidines, and you can see that they only have a single ring structure. RNA is the translation product from DNA, and it is comprised as a single backbone structure, and the nucleotides are the same as for DNA, with the exception that there's no thymine in RNA. Instead, there is a uracil. Most RNA exists in shorter strings, or shorter strands, I should say, than DNA is found.
So information in the DNA gets decoded and translated into proteins, which then perform the work of the organism at the cellular level. So in order to decode this information from the DNA, it's largely occurring in steps. And I'm sure you're familiar with this, so we were just going to review it here. The first step would be transcription, which would be converting the DNA into messenger RNA. So we have the DNA here located within the cell's nucleus. We can tell this because we see here our cell membrane, so the DNA is here within the cell nucleus. So first step here is that it's transcribed into mRNA. And once that mature mRNA is formed, it is now leaving the cell's nucleus, going into the cytoplasm, where it is it's now translated in the cell's ribosome. So in other words, in the translation here, the mRNA is directing the assembly of amino acids into protein molecules per the instructions that it got from the DNA. And then the last step, the third step, is translocation. So that means now moving this protein out of the ribosome to wherever it's needed elsewhere in the cell. So these proteins that are being synthesized, they have various roles. Earlier I mentioned enzymes, but they could also be receptors, transporters, antibodies. They could be functioning as a hormone. They could be functioning in cell communication. So much of the work that's being done within cells and within our body is being done by proteins. Another important role that's important for understanding genetics is the role of transcription factors. So transcription factors are biological components. So this could be a hormone, it could be a vitamin, in particular vitamin D and vitamin A are important transcription factors that bind to the 5' end of the gene and help increase or decrease transcription of DNA into messenger RNA. So these transcription factors, they're not changing the DNA, okay? The instructions are always going to be the same, but they might be upregulating the translation into mRNA. And since the mRNA is what's actually going to make the proteins, that's what's really important, right? So I guess you could think of it like the DNA is like the instructions, but the mRNA is going to be the person who reads the instructions and tells the other people what to do, if you're thinking of maybe running a little business here. So it doesn't matter if there's instructions and nobody reads them, right? You need somebody to read it and then do it. So as I mentioned, bioactive food components, so things like genistein found in soy, some vitamins, polyunsaturated fatty acids, these all have roles in gene transcription. They can bind to these transcription factors and thereby influence how a gene is expressed. And so this would be an example of nutrigenomics. In the field of genetics, you will hear a lot about gene mutations. A gene mutation is simply a change in the DNA sequence that can alter the amino acid sequence of a protein. It doesn't always alter it, but it can alter it. If the change alters the function of the protein significantly, this could be an instance where disease risk could increase. And so we refer to things as gene variants as the gene that has a slight variation in its code from the normal gene, which is going to have an impact on the function of the organism. So gene variants aren't always a bad thing. Sometimes it might be enhancing production of a protein that's beneficial. On the other hand, it could be something that's deleterious to health, producing maybe a protein, downregulating the production of a protein. So one of the variant forms of a gene at a particular locus on a chromosome is referred to as the allele or the allelic variation. So you will recall that I said that you have 22 pairs of autonomous chromosomes and 2 sex chromosomes. And so alleles are referring to the variation of a gene in the same place on the same chromosome. So you've got a pair of alleles, one from each parent. If they're different, then you have your allelic variation. If they're the same, then you don't have variation. So because of the Human Genome Project, and we know what the typical variation is, if one's allelic variation is different from what we typically see, that's called the mutant allele. Although mutant sounds like a bad thing, again, it could be that the mutant allele actually promotes your health, or it could be in the opposite direction. So you don't want to necessarily think that having a mutant is necessarily a bad thing. Okay, so each pair of alleles represents the genotype of a specific gene. So a genotype would be homozygous if there are two identical alleles at a particular locus, and they're heterozygous if they are different. So again, what we're seeing here in this diagram, this would be heterozygous, right? Because we have a mutant allele and one normal allele. In addition, some alleles are dominant or recessive. So when an organism is heterozygous at a specific location and carries one dominant and one recessive allele, the organism would express the dominant phenotype. So the phenotype is what is presented. Alleles can also refer to minor DNA sequence variations between alleles that did not necessarily influence the gene's phenotype. And this can be related to Mendelian inheritance. So the genotype is an individual's unique genetic makeup. We all have our own genotype. Our phenotype is the measurable expression of the gene. So this phenotype is just simply says it's what presents itself. It's how you look. So you're homozygous if you have two identical alleles at a genetic locus, and you would be heterozygous if you have two different alleles at the same genetic locus. I referred to the phenotype as the example of a physical trait of what you look like, but it also could refer to the level of a protein found in your blood. So a little bit more here on Mendelian inheritance. There are 5 classical modes of inheritance: autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and Y-linked. So the autosomal dominant and recessive are referring to the genes that are located on those 22 pairs of autonomous chromosomes. Autosomal dominant is referring to genes with characteristics that are expressed only when a single copy of an allele is present. So if that's an autosomal dominant trait and you have just one copy of that dominant, then you would express it. The recessive occurs when the changed allele is present, but it's not expressed unless both copies are present. So if you think back to your high school genetics where you learned about Gregor Mendel and you did those little 4x4 and the 2x2 plots, the X-linked recessive would be in that bottom corner where you have 2 of the lowercase letters for the recessive genes. So some examples of autosomal dominant diseases would be things like Huntington's disease, which is the degeneration of certain nerve cells in the brain resulting in dementia. Familial hypercholesteremia, in which individuals have defective LDL receptors being synthesized in the liver and ultimately end up with very high LDL cholesterol. Marfan syndrome would be another example of an autosomal dominant genetic disease. Autosomal recessive diseases include many of the known inborn error of metabolism. So PKU, for example, is an autosomal recessive genetic disease. Cystic fibrosis and sickle cell anemia are other examples. The X-linked dominant diseases usually occur in males. And overall, more of the X-linked diseases occur in males. And this is because males only have one copy of the X chromosome. So a single recessive gene on that X chromosome will cause the disease. The Y chromosome, as you know, is the other half. So the primary nucleotide sequence of the genome is about 99.9% identical among all humans. So this 1.1% variation between humans is what constitutes the molecular basis for most human phenotypic variation. So the differences between Okay, so let's talk a little bit about nutritional genomics. Currently, RDs already play a role in genetics by providing medical nutrition therapy for chromosomal and single gene mutations. But I anticipate that in the course of your careers that this role is going to expand quite a bit. So this slide here is showing us a little bit about B vitamin metabolism. And in particular, we're going to talk here first about an example of nutrigenetics. So folic acid, through its metabolism, becomes tetrahydrofolate, and then it then gains a methyl group to become 5,10-methylene tetrahydrofolate. It actually gains 2 methyl groups here. But then an enzyme called methylene tetrahydrofolate reductase, this MTHFR, this enzyme removes one of these methyl groups to make 5-methylene tetrahydrofolate, which is necessary for the conversion of homocysteine to methionine, and ultimately the production of acetyldecyl methionine, which is important for the formation of new nucleotides. So what I just discussed to you now, the specifics of that are for metabolism class, not for what we're talking about today. But it is not an uncommon genetic variation that individuals have decreased production, their genes code for decreased production of this methylene tetrahydrofolate reductase. So when an individual has a SNP in their genetic code that creates less production of this enzyme here, what happens is that they don't have enough of this enzyme to have the 5-methyl tetrahydrofolate form from the folic acid. And they end up with increased homocysteine in the blood, which we know is a risk factor for cardiovascular disease, and then also insufficient production of acetyldecyl methionine to go on to create for the synthesis of nucleic acids. This is a fairly common variation that occurs for the substitution of a thymine for cytosine at site 677 in the DNA. They end up producing an enzyme that's a little funky of this methyl tetrahydrofolate reductase that's thermolabile. It means it's very it's very sensitive to heat, and so much of it which is produced ends up lysing and not being able to function properly. And so ultimately they end up with reduced activity of this enzyme. So the tailored nutrition therapy for an individual like this would be to supplement with folate, B6, and or B12, because all of these B vitamins are related in folic acid metabolism and have interplay with each other. and then to monitor the levels of homocysteine in the blood to see if you could get them on a normal level. So this would be an example of nutrigenetics. So how the gene is influencing, or the genetic code, how the genetic code is influencing an individual's specific nutrient needs. On the other hand, we have nutrigenomics, or how what we're eating influences our genes. So bioactive food components, which include some vitamins, as I mentioned earlier, influence the activity of transcription factors on DNA. So these have a variety of modes of action. It could be signal transduction. So in other words, cells have cell signals on the surface of the cell that can sense what's going on in other areas of the body, which will then send a transcription factor to the DNA to either increase or decrease translation of certain proteins. Another important role of these bioactive food components and vitamins is to serve as ligands. So, for example, sometimes a ligand is needed to activate transcription of DNA. So let me give you an example here for omega-3 fatty acids. So omega-3 fatty acids serve as precursors for the synthesis of anti-inflammatory eicosanoids. So they also help to decrease expression of genes that lead to inflammatory cytokine production. So omega-3s as well as omega-6s have been found to serve as ligands for transcription factors, which help upregulate lipid and lipoprotein synthesis. And I think I have a picture of this on the next page. Let's take a look. Okay, so ligands are things which are going to activate transcription of DNA. I don't have a picture of it here, but you can think of, you've probably learned about ligands in kind of like a lock and key model, that the ligand kind of clicks into a part of a key in a diagram that allows it to be turned, which then will allow for the transcription to occur. So amino acids, fatty acids, folic acid, B12, fat-soluble vitamins, they've all had roles as, they've all been identified to have a role in gene transcription. So within the promoter region of the DNA, these vitamins and bioactive food components help to turn on the transcription of the DNA to make the RNA, which ultimately will go to the ribosome and create new proteins. In the previous slide, I also talked a little bit about signal transduction. So some nutrients will activate transmembrane receptors, which will then use intracellular signaling pathways to initiate or modify gene expression. So mostly this is amino acids and fatty acids, which will work through signal transduction. So you can just think of this simply as these signal transducers on the cell membrane. They can sense the fatty acid or the amino acid, and then it sends a message to the DNA to upregulate transcription. So chronic diseases are thought to result from a combination of genetic and environmental factors. Overall, chronic diseases to study and to treat and to understand and prevent are more complex than single gene disorders. That's not to say that single gene disorders don't have a profound implication on the people who have them, but the treatment is often a little more clear-cut than it is for a chronic disease. So it's thought that most chronic diseases have multiple genes involved. And chronic diseases are more likely to be influenced by environmental factors, so things like stress or exposure to chemicals. Gene variants predispose an individual to chronic diseases, but nutrition and lifestyle factors are often affecting the expression of the gene variants. So when we talked about nutrigenetics and the examples of folate metabolism, for example, that a single SNP is affecting folate metabolism. On the other hand, nutrigenomics is likely in what you eat, is likely affecting many different genes and their gene expression. And I would say, in my opinion, that nutrigenomics is probably having the greater effect on chronic disease than nutrigenetics. On top of this, because we all have variation in our DNA, we are all responding differently to environmental influences, including what we eat. So some individuals have more of a response to a dietary treatment than others. And increased research and knowledge in nutritional genomics may allow individuals to have more personalized nutrition care plans that are based on their genetics. So a little bit more about the application of nutrigenomics with chronic disease, specifically cardiovascular disease. We know that there's a high degree of variability in response to traditional medical nutrition therapy for hyperlipidemia. So, for example, some people who follow traditional MNT to lower their LDL, it ends up that their triglycerides end up going up and their HDL cholesterol goes down, which is not what you want. And some people have a dramatic response to soluble fiber, where others have a more modest response. It appears that medical nutrition therapy should be tailored to the patient's genotype for the best lipid-lowering effects. There's evidence to suggest that variations in ApoA1, ApoA4, ApoB, and ApoB genes contribute to the heterogeneity in the lipid response for a dietary intervention. So specifically here, when we look at the ApoA1 gene, codes for the apolipoprotein A1, which is the primary protein in HDL. And there's a variant that's been identified in which the guanine has been replaced for an adenine at position 75 in a regulatory region of the ApoA1 gene. And so for individuals with a normal allele, increasing polyunsaturated fatty acids results in a decline in HDL. And women with the altered allele had increased levels of HDL when PUFAs were increased. So this is particularly relevant, especially since the TLC diet recommends up to 10% of total calories from PUFAs. So nutritional genomics research works on identifying, noticed a typo here, sorry about that, identifying gene disease associations, the dietary components that influence these associations, the mechanism by which dietary components exert their effects, and the genotypes that benefit most from particular diet therapy. Expectations for nutrigenomics is high, but the progress so far has been pretty slow, and interest is truly greater than the current research. Some of the challenges to meeting the expectations have been that a lot of genetic work is expensive, although it has decreased, the cost has decreased significantly in the last 10 years. It can be difficult to obtain tissue samples from healthy volunteers to do genetic analyses. This is for a variety of reasons. Generally, it's more difficult to do any type of research that's requiring a tissue sample or a biological sample. And also, some healthy volunteers are not willing to submit samples if they know they're being used for genetic profiling. There's ethical issues in terms of maintaining patients' confidentiality. Many patients and consumers are worried about what would happen if their genetic information were revealed to health insurers, for example. In 2008, the Genetic Information Non-Discrimination Act, or the GINA, was passed, and it prohibits health insurers
Thank you everybody for your attention. This concludes my presentation. If you have any questions, feel free to send me an email or pass those questions on to Professor K, and she will make sure that they get to me. Thank you.